good to record. Thank you.
Thank you,
Sure.
Vero.
Yeah. And so as Veronica said, we started working on this a couple of years ago, and at the time we had a protocol, a very general protocol for preparing extracts that it was one among many, and some of you probably know more about the preparation of extracts than I do, so please feel free to intervene. And I hope that this is more of a discussion than in an interaction than me just speaking. But it's a fact that different labs use different protocols to prepare extracts, and also protocols to prepare the extracts vary according to the organisms that you're studying. And what we someone has the microphone on. Okay, it's off now. And what we thought was that maybe it would be good that since there's more and more people interested in preparing extracts, either for their own research or to collaborate with labs that can use them in a variety of assays. What we thought was that everyone should have sort of the same protocol, and that protocol should be rational, and it should be relatively easy that everyone could do it, so that's the purpose of the discussion today. And to do that, as Veronica said, a month ago, I went to the National Cancer Institute, who has been generating one of the largest libraries of natural product extracts in the world. And I met with Barry O'Keefe, the director, to see how they do it. And because if they probably had to go through these same questions, and if they have a protocol that works for them, maybe it also works for everyone else. So that's going to be the focus. But I'd be happy if any of you actually, if already preparing extracts, would be able to share their knowledge. And so the National Cancer Institute program for natural product discovery deals with a variety of organisms, and they have different protocols to prepare extracts depending on the organisms are plants or marine or microorganisms. So we'll go over that. And the idea, as I said, is to have a protocol that that makes sense, that is similar to, in case you feel like you want to adopt it, but it's similar to everyone else, and that people can use to explore new sources of natural products for their research or their collaborations. So I'm going to present first some slides that some of you already know on the process of developing therapeutics from natural products. But I'm going to go over them because some of you don't know, and because it will help place in context the preparation of extracts. So what's the interest in natural products? And many natural products are a rich source of chemical diversity, and this is really important in chemistry and when you're looking for drugs. In addition to that, a lot of these compounds actually are biologically active, as they have been evolutionarily selected to work in a cellular environment, and many times to actually target processes in the natural products that are also relevant in the context of human disease. There are many sources of natural compounds. You can find them in libraries, commercially available or governmental, just like the case of the NCI. You can work together with traditional medicine practitioners, with biologists, ethnopharmacologists, or you can find them even in collections of fungi and plants. How do you select natural products for your research? Well, you can take an unbiased approach and just study all of them. If you have resources to do that, maybe that would be the best. But in most cases, we don't. So you have to select them based on some sort of criteria. And one criterion is, the published literature on ethnopharmacological use of the natural product. So once you select what you want to study, you have to prepare an extract, and this is going to be the focus of the discussion today. And most often you dry it and you grind it. You have to choose a solvent to extract the metabolites in the natural product, and this is one of the most critical aspects, because a solvent has a certain polarity, and that's going to extract a different array of compounds. Then there are several methods to do the extraction, and at the end you have to filter, dry the extract, and resuspend in a solvent that is compatible with your screening. So if you do a screening before you test in animals, which is usually what people do, you want to have a screening that is easy and inexpensive, so that you can first test a large amount of extracts or compounds, right? And there's two types of screening. You can have a forward pharmacology screening approach, also called phenotypic screening, where your screening assays are biological and they represent phenotypes, and you are going to identify extracts or compounds that protect in those assays, and then only after that, after you know that they work, you're going to identify the targets. But you can also have screening assays that are based on a reverse pharmacology approach, and in this case, you already need to know what to target, because that's what you're going to mimic in your in vitro assays. And then only after then you test in the biological models, right? So, and then you can, once you have something that works, an extract that works, you can try to identify the biological active compound, or you can just work with the extract. So that depends of your objectives, right? So this only applies if you want to find out the active compound. There's many ways of doing that, which we will not talk about. And then if you do have a compound, and if you want to optimize it through medicinal chemistry, you can do that. But anyway, regardless of that, you will end up testing either the compound of the extract in an animal model of the disease or the condition that you study. There are many species available. You can look at things like behavior, biochemical markers of the disease, or you can even use omics to understand how your extracts or your compounds are working, and what's their effect in the disease model. So once you do this, at least in the US, you have to apply for an investigational new drug, which requires heavy safety and toxicology studies. And if that gets approved by the FDA, then you are allowed to carry out a clinical trial in humans. So basically, this is the summary of the process. Regarding the extraction of the preparation of extracts, so that you can do your research. So as I mentioned, most of the presentation is going to be focused on what the people at NCI is doing. And they started their program in 2019, although, or it was launched in 2019, but they started preparing extracts back in the 80s. It's one of the largest and most diverse collection of natural product extracts in the world that is available for high throughput screening. In addition, they have also produced fractions of the extract. So it's a pre-fractionated library. And I will explain this better by the end of the presentation. What are the advantages of having a pre-fractionated library? It's available to everyone. It's free of charge, and you can use it to test you can use it to study any diseases, to develop therapeutics for any disease. It's applied in 384 well plates, both with fractions and the crude extracts. So they assembled their library through letters of collection that they have established with countries all over the world in these places where you can see here in the map, including the US, evidently. And these letters of collection actually predate the Convention on Biological Diversity, although they already stipulated equitable benefit sharing in case anything would be made commercial out of their research. And so the library comprises over 100,000 different natural products. And as I said, they are preparing fractions to supply with them. And they actually, as of today, they are still preparing the fractions because the library, once it's finished, is going to be supplied with 1 million fractions. So you do have to pay the shipping freight, but the rest is for free. So this is just to give you a background for their operation, right? Okay, so a lot of the things that I'm going to say, you can find them in these publications, and either you can access this through the movie, I don't know, Veronica, or I'm happy to share the slides as well. But they are their own publications from the NCI, and I recommend reading them. And what I'm going to talk about, a lot of it is the information is contained there, right? So as I mentioned, they prepare extracts from plants, from marine organisms, and from microorganisms. And they prepare both aqueous and organic extracts. Now, the way of preparing these extracts varies according to the type of organism for specific reasons that I'm going to explain. The plant, so the plant specimens, which are over 80,000, are the extracts of the plants are prepared by starting actually with an organic extract extraction using 1-to-1 of the chloromethane and methanol, and then they do the water extraction. For the marine organisms, they start first with water so that they can remove salts and polysaccharides. I'm going to explain you all this after the slide. And then they do a 1-to-1 organic solvent extraction in the chloromethane and methanol. And for the microorganisms, which are fungi and bacteria, the extracts are prepared by homogenizing cultures that are grown in vitro with 10% methanol, and then followed by the addition of 1-to-1 of the chloromethane, and then by partition of the solvents. Okay, so regarding the plants, just a few notes on the collection step. And also before that, so this protocol, it can also be applied to dried lichens and mature fungal basidiocarps, as long as everything is dried. So just to remind you that for collecting any natural products that you study, people need to obtain the required local permits to do identification of those samples, and very importantly, any material should be deposited in an institution. So I'm talking about voucher specimens, and this is very important. So you should also keep track of the location where you collect your material, the date of collection, and any other information associated with it, traditional uses, etc. You can collect any part of the plant or the material and prepare specimens of that. And the material has to be dried, and this is so that you avoid having organisms growing in there and interfering with your study. At NCI, they have quarantine import stations. So when they get the material, it's kept sealed at -20 for several months. And I believe they do something similar at the Harvard area when they get plants to add them to their collection for the same reason. Okay, and so once you have, let's see this. Once you have the material, you have to grind it, and for that they use an industrial grinder, literally, that is placed inside a chemical fume hood. So they're very concerned about protecting the people that do the preparation of the extracts. Bear in mind that they do this to thousands of specimens, and some of them, you don't really know what you're dealing with, right? So some of them may have irritants, and people have to take the adequate precautions. You should also avoid cross-contamination, evidently, and how they do this, how the specifics on how they clean the material, you can find them in the publication. I'm not going to go over that. But after grinding, what they end up having is particles that are in the 0.5 to 2 millimeter size range. Okay, so once you have your material ground, what you do is you start the extraction. And they have tested many different extraction techniques. A lot of them, they were considering practical for a large-scale preparation of extract, right? And they also tested lots of different solvents. And the idea was to come up with something that was inexpensive, that could be done at a large scale, would take up less time, less space, less resources, less equipment, but at the same time would generate what needs to be generated, and it would produce an extract in a rational way that can be representative of the metabolites of the material where it comes from, right? And if they did this, I think these are all criteria that we also have, right? So we could learn from them. So as I was saying, they tested. This is just an example of an extraction that they did with several different solvents and of a plant, and where they see that where they get the higher amount of extract yield is when they do a mix, when they utilize a solvent that is a mixture of dichloromethane and methanol. And they reason that what's happening is that the DCM and the methanol are really good at dissolving the plant matrix and making the compounds available for the extraction. So the methanol is a very good solvent. It has an intermediate polarity and is relatively inexpensive. And importantly, it can be dried out very easily using a rotovap in the lab. So, and this is important because you'll always have to remove the solvent from your extract because it can be toxic for your cells or it can interfere with your assays. And so you have to think about this, right? And ethanol, which is the most usually, probably one of the most used solvents in labs all over the world, because it's it's cheaper, it's easier to obtain than methanol. however, has the disadvantage of being much harder to dry. And in addition, in their sample, so this is just an example, but this is representative of what they saw. Actually, it didn't produce as good as an extract as the DCM and the methanol. Now, this is gonna depend on your plant, right? So they also tested different amounts of incubation of the material with the solvents. And what they saw was that with time it increases, and after a while it doesn't become so relevant. So they ended up choosing a time of extraction that is associated with being practical. So they just leave it overnight, right? Because they don't want people working weird hours without necessity. So they just leave it for 15 hours overnight, and then when people come the next day, they can continue the process. They've also studied the effect of doing sequential extractions with the same mixture of solvent, dichloromethane and methanol. And what they saw is that most of it comes out in the first pass. So if you re-extract with the same solvent, you don't see much of a difference. They've also tested methanol, and I will explain you why. And what they also see is that after this, when you extract with water, again, you can extract more compounds, right? And this is the basis for the reason why they do a sequential extraction with water at the end. And the idea is that in the dichloromethane, removes a lot of these lipids of the plant matrix that makes the compounds available then for methanol and water to be extracted. Okay, so what is the protocol exactly? So they start by placing the ground material in a percolator, and then they add dichloromethane and methanol 1:1 until they cover all the material plus an additional volume, because the material tends to swell, right? Then they cover it and they steep overnight, as I said, at room temperature in the dark inside a chemical fume hood. Then the extract, so they remove because it's a percolator, right? They remove that extract, and then they add methanol, and then they soak it for 30 minutes. And the idea of adding methanol here, which is then combined again with the dichloromethane and the methanol. And the idea is that not only it increases maybe a little bit the extraction yield, but not much, as we saw, but it also removes the dichloromethane in the material, which is toxic. And so okay, so once you have all that mixture of extract, you put it in a rotovap to evaporate it. Eventually, so it starts in these big bottles, then they transfer it to smaller flasks, and they dry it completely, and they freeze it at -20, right? So all the organic solvent waste is discarded through a contractor. I don't know how this would be worked out in your institutions. They don't do any recycling with distillation. Okay, so once you do this, you have the plant material left over, which goes straight into water for the preparation of the aqueous extract. So they just had high purity water until they cover all the material. It steeps overnight, and then they remove it again, and then they lyophilize it at -40 in specific freezers. And the reason why they don't do the rotovap is because sometimes you have to heat it, and because the compounds are in water, they don't want to accelerate hydrolysis and the decomposition of the metabolites. So they're very careful about everything. The plant leftovers, they just incinerate them. And then, yeah, the washing, it's all detailed in the publications. So no need to say that you have to keep record of every single weight throughout the process, because you need to calculate the percent yield, which is required for publications, and also to know how efficient is your extraction, right? And this is a table with the average percent yields of the organic solvent and aqueous extracts, average from all the different parts and all the different specimens that they use in the library. Just have an idea. So most clinically used molecules are going to be found in the organic solvent extract, right? Now, I wonder what is the relevance of the water extract, because we have tested water extracts in our lab, and they are not nearly as interesting in terms of positive hits in our assays as the organic solvent extracts. So it could be that you have less amount of compounds. It could be the type of compounds that you have. They're going to be more polar. I'm not sure. We can discuss this at the end, and maybe you can share your knowledge. They also, just out of curiosity, they looked at the survival of microbes in their extracts, which is very unlikely to have any in the dichloromethane and the methanol extract. And the ones in the water extract, they actually tested them and they grew very slowly. They just did this because they were concerned that that could affect their studies, having other organisms in there, right? They looking at chemicals. Okay, so this is the protocol regarding the preparation of plant extracts. For the preparation of marine extracts, and these include marine invertebrate animals and all sorts of algae. So what they do is, and I wonder if you can also do, if you could dry the algae and do the other protocol, but they utilize this. I guess it's because of the salts that you sometimes get and other things. But the samples are quickly frozen upon collection, wherever they're being collected, and then they are shipped on dry ice to NCI, right? And they are kept frozen at -20. So they want to minimize any alteration in the material, and namely the degradation of molecules, right? And even the extraction is done as much as possible in the cold, as you'll see. And so the purpose of doing the aqueous extraction first is to remove the salts and polysaccharides, which would eventually affect your extract, your organic solvent extract, if you were to do an organic solvent extraction first. So they did try to do an organic extraction first, and they saw no specific benefit. And what happened actually was that when they went on doing a water extract afterwards, it would form a gel, which would make it, this is what they claim, would make it really difficult to process. Okay, so how do you grind these things? Because many of these organisms have shells, which contain irritating silicate spicules. People have take all the precautions, and they're careful not to get in contact with them. Then they do set aside lyophilized material, ground material, as a voucher, so for taxonomy. And so what they start is by mixing the material with dry ice. And the idea is to make the material very cold so that it's also very brittle, because they're going to put this through a grinder, right? And for things, harder specimens like corals and bivalves, they even use an hydraulic press to destroy the material, right? And they grind all this in a hamburger grinder to generate pieces that are about 1 inch in size. And after this, they after grinding, they put all this in the freezer so that they can sublime the dry ice, right? Because it was all kept as much as possible, right, in frozen, right? And then they have to remove the dry ice, and that takes a few days. Okay, so then the material is ready for the aqueous extraction, and what they do is they put the material, they cover the material with water 3 to 1, and they keep this at 4 degrees for approximately 45 minutes. And then they go on centrifuging all the samples, and then they lyophilize the aqueous extract, right? And they also lyophilize the material, so they want to remove the water out of the material before going into the organic solvent extraction, which you do basically by re-extracting with the same mixture of the chloromethane and methanol that you use for the plants. So they do this in a percolator as well. And after soaking overnight, so the 15 hours, the material is drained and washed with methanol, soaked for 30 minutes, and then mixed with the dichloromethane and methanol extract. Goes to the rotovap and is completely dried, and then it's ready to be stored at -20. So again, everything needs to be weighted. These are the extract yields. I just included them here for reference that you can use if you're already doing this, or if you'll get to do this, and to see how much you get and maybe what you should be getting. So since there are essentially no salts in the organic solvent, so these extracts are rich in somewhat polar and non-polar molecules, right? Because of the methanol and the DCM. And again, just like with the plants, the organic extract is the one that is most likely to have metabolites that have drug-like properties. Okay, so how do you proceed with the extraction with the microorganisms? And we're talking about fungi and bacteria. So they grow them in fermentation in culture for 1 to 2 weeks. And they've optimized the culture media. Now they use the same formula, for example, for bacteria. They said that they are using the same formula for any bacteria. So that means if you change the media composition, you're probably going to have different metabolite compositions. Yes, but they cannot test this with thousands of species, right? So they just assume it as it is. They do prepare the extracts in those conditions, and they have the organisms, they have the microorganisms frozen in case someone wants to play around with the conditions and then do that on their own, right? So into harvest, after you grow them in culture, you just add 10% of the volume in methanol, and then you homogenize it in a homogenizer like this to disrupt the cells. Then you mix it with the chloromethane 1 to 1. And you shake it, and you wait for the layers to separate. And because the chloromethane is going to be in the bottom, it's removed first, and that will be the organic extract, which are going to be dried in a rotovap, just like in the other protocols. The aqueous fraction is going to go through filter paper because it's going to contain all the cell debris, which is then lyophilized at -40, and everything is stored at -20 and ready to test. So that's pretty much it. So I've put together I've assembled some final considerations, ideas that I thought they were interesting to maybe stimulate things to take into account and stimulate the discussion. So one is, again, the attention to voucher specimens. The reason why people prepare voucher specimens is, or before that, so the voucher specimens are collected at the same time and from the same lot of the material that you're studying. And there's a really good review on this that you can also go over it later. But the idea is to preserve a record of the original sample tested so that anyone can go back to it and analyze it. And in addition, you're also providing a lasting material to be accessed in the future, right? Now, there are several considerations, right? Because depending on the labs, you might not have access to many of these resources, namely instruments. So I guess you'll have to adapt as much as possible. I mean, shredding and most of the stuff that they use is actually pretty rudimentary. So I think it can easily be done. I think the most critical is going to be the solvent, maybe. Some people might not have access to dichloromethane or might rather use something else because it's toxic, and maybe some institutions are not happy with that. And I don't have a clear answer to that, so maybe people can share, but perhaps you can do an extraction just with methanol and then water, or combine them. If we look at the sample that they gave us, you can see that I got this. That the methanol was actually, alone was better than the ethanol. They have a mixture of methanol in water and methanol 9:1. I don't know what is the most common when you have both of them together. But if you should still do water, because clearly there's a lot of things that are not going to be extracted by the methanol. And I guess the important thing is to make the metabolites available, take them out of the cells and make them available to the solvent. But we can discuss this. So other considerations that you should have is whether there is enough material, right? And this is important not from the perspective of sustainability, and in some species. You might not have lots of them and lots of individual organisms, but you should think about that. If everything goes well and some of these things turned out to be positive in your assays, that you might need to harvest and collect more of your material. Especially if you want to identify the compound, an active compound underlying the activity, or even more if you want to test it in animals. And this applies to all extracts, right? Even if you don't go into identifying active compounds. Now, there is an advantage of working with microorganisms, right? Is that you can grow them in your lab, in culture, indefinitely. You can make as much as you want without having to intervene in the environment, right? You collect them and then you grow them. You can freeze them, and people that work on metabolites from microorganisms, whether in the therapeutic discovery path, they like them because of this, right? Also, if you're working with natural products that are used in traditional medicine, so there's also the consideration of how the traditional medicine is prepared historically, right? And so what they do at the NCI, which makes sense, is if someone asks them to test something or to prepare an extract from something that is used in traditional medicine, because in addition to their program on natural natural product discovery, they also do therapeutic development for cancer, right? That's what the NCI stands for. So when someone asks them to do that, what they do is they prepare an extract using the same method as it would be prepared traditionally. And part of the material, other half, they prepare an extract using the method. And now using the traditional method is important for several reasons, or maybe relevant for several reasons. And one is that many journals, especially those specific with a focus on ethnopharmacology, are gonna these days they require you to study the traditional natural product that is used in traditional medicine, using the same traditional method, right? So that you can enlighten on that ethnopharmacology. Now, from a drug discovery perspective, you just want to maximize the extraction yield, right? And water, which is mostly used in traditional medicine to prepare infusions, etc., is not a great solvent, especially if you just use it without solubilizing all that material. And finally, just going back to this idea of preparing fractions of your samples, I know that this probably requires technology that most people don't have, but if you do, you might want to consider doing that. And all the details on how to prepare the extract, so the type of HPLC column and solvent and gradient that they use, it's all published, so you can have access to them. And there are advantages associated with preparing fractions. And for those who don't know, fractions are just different. It's just different parts of different combinations of the metabolites that you have in your whole extract that are separated, usually through a polarity gradient. So you get compounds separated from each other in different fractions. And that means that in each fraction, you have less compounds, and you have more of those compounds than you would find them in your whole extract. And this has several advantages, right? And so one of them is that it increases your screening performance because you have less material in there interfering with your screening, especially if you're doing target-based screening. And also another advantage is that because these fractions have higher amounts of compounds that could be in minor amounts in the original extract, you increase the chances of picking up compounds that are active but that exist in lower amounts, right? Also, because these fractions are different and prepared according to a polarity, sometimes there are specific classes of metabolites that are associated with interfering with specific in vitro assays. And what happens is that they end up all being contained in specific fractions. So the other fractions are cleaner, if that's an issue. And also, doing fractions also helps with dereplication. So they do analyze all those fractions by mass spectrometry. So if you see something positive, you can see what's in there, and if you already know that some specific compounds are active in your assays, then you might not have to go and identify the compounds in those fractions. But because these fractions contain a lot less compounds than the original extract, they make it a lot easier to do the compound isolation. And the way they do this is if someone finds a fraction that is active in an assay, they go on preparing 20 more fractions of that fraction. And usually that's enough to identify the active compound immediately. So anyways, just this, but this already requires additional technology. And so that's it.
Thank you very much, Antonio.
Sure.
Yeah, this is really interesting. So I would like to invite other people who are experts on this, who work every day preparing extracts, like the people in Panama, for example, if they can make some comments. Yeah, Marcelino, are you there?
Yes, I'm here.
Yes, if you can give us your thoughts.
Sure, it is very interesting. Thank you, Antonio, for sharing all these protocols. I think I'm sure there is plenty of minds behind these protocols. So we do something similarly, we do some things differently, and it's good that you share and compare different methodologies to see which work better. So I think for this group, probably we need to write a protocol. So everybody start using the same protocol to prepare extracts. I think like a written document would be good. And also we need to consider, in our case, we work more with microorganisms that are grown in solid media. So when we're using solid media, extraction is different. So what we do is we lyophilize and then extract with solvents, organic solvents. But I think that for liquid media, the protocol that you show is very good.
So how do you do it with the solid media? You scrape the microorganisms, or you dry everything?
No, we have the agar plates, and then we split it in small pieces and put it in a lyophilizer, and then we extract that with ethyl acetate. And that's the way we do it. But you can use methanol, like chloromethane also. Probably that will work better. But we lyophilize to eliminate the water. We put in. So this is what we do. I think we learned these protocols in the Bill Gerwick's laboratory and Bill Fennichel's laboratory at Scripps Institution of Oceanography
in San Diego.
So we standardized that in Panama. But this is very good to know what NCI used, because I think there's a lot of minds behind these protocols. I think we have to consider that.
Yeah, exactly. That's the reason, Antonio, visit them. Anyone else? Christian, do you want to add something? Who else is preparing extracts in this group? What about Costa Rica? Is anyone from Costa Rica? I think they...
Hello.
Yeah, hello.
Good morning. It was a really nice presentation, but I had a question regarding the type of solvents that were used for the extraction of the different kind of materials, right? There was a heavy use of dichloromethane, but I was wondering if there is a sort of alternative that could be used, because recently the FDA, for example, stated that they are discouraging the usage of dichloromethane because of the heavy impact that it could have on the environment. So I was wondering if there is something similar that could be used, so that it could be, because the FDA is disregarding the uses of this kind of fluorinated solvent.
Excuse me, Randy. Can you introduce yourself? Who, where?
Yeah. Sorry, I had a microphone issue at at the beginning of the session. Okay, my name is Randy Mojica. I work with the group of Dr. Marcelino Gutierrez here in Panama at Indicasat. Currently, we are working with several kind of extracts, mainly from fungi. There are collaborators of us that are working with bacteria. So we currently are not using that much of the chloromethane because of the cost, because of the environmental users, and we try to be more green in that aspect. So I was wondering why the heavy usage of the chloromethane in in the examples stated.
Thank you.
Yeah, so that was actually the point of addressing that in that last slide, that last couple of slides. And maybe someone has a better explanation, and I do, but my guess is that if you want to avoid it, then I would go for methanol, right? And some people just prepare extracts with ethanol. But I think it's the kind of thing that is good to discuss with everyone, right? So that everyone is on the same page. I don't know if anyone wants to add something.
Good morning, Antonio. I'm Christopher from Marcelino's group. Also, I just wondering in this tech that you do, that you always use a mixture of solvents. For example, here in Panama, what we usually notice is that the solvent we prefer to do a stack extraction with a polar solvent and a high polar solvent in order to avoid the complexity of the sample. Since if you do a mixture of solvent, you actually getting a low complex extraction, and sometimes the activity is being high due to the sugar part of the compounds that you've been extracting with this methanol extraction. Could you?
Yeah, sorry. Yeah, again, I'm not a chemist, but I know that some people do choose to do sequential extractions of of solvents with different polarities, right? And my understanding is that they're because they're dealing with thousands of extractions, they wanted to optimize the process. So I guess the idea of using DCM with methanol is that the DCM does break up the material so that they can do the extraction with the methanol. And I think most of the metabolites that may be more interesting from the drug point of view are going to be in that fraction. And so maybe by doing that, they saved on time and money of adding steps for preparing more extracts that maybe wouldn't be required. But I can't answer for sure. Yeah.
Thank you.
Nobody else wants to make any comments on this? What about Peru? I think this Miguel Angel's colleague. I don't have her name right now.
Hello, Lena.
Yes, Lena, yes. Can you make any comment? Because I understand you also work on this, right?
Well,
Not really.
we
Yes.
will just start working on traditional medicinal plants from our Andean region.
Yeah.
But we have only experience in extraction of secondary metabolites. So we apply basically an targeted metabolomic using specific solvent mixtures based on aqueous, ethanolic or methanolic and acetone extracts. And that's what we do, and then we evaluate different in vitro models for screening the functionality of these extracts. But so far we have more experience in food-derived extracts from our diversity. Now we want to start to screening the potential of our medicinal plant, traditional medicinal plants diversity. So this workshop is key for this task.
Excellent. So we are recording, so you can take a look again, yeah? And of course,
Sure,
Antonio is open to respond to all your comments and questions, yeah? Anything else that you want to add, Antonio?
But no, I mean, again, I'm not an expert on this, right? But I think the key here is to to throw the discussion out, because I mean, from even just looking at it, why do people have different protocols? That doesn't make sense, right? If one protocol or some protocols make more sense to use, why is that people are doing things differently? So, and this was was the departing point for going into this and have this discussion, especially if people are going to be collaborating with each others, and we thought that it would make sense to have some sort of standard protocol that could be executed, that could be carried out in most labs, let's put it that way, that wouldn't require sophisticated technology that only some people have access to, and that would be rational. And by rational, I mean that it would be efficient to extract compounds that then you can or metabolize that then you can use in your research, right? So I think that was the main goal.
This is Pam Maher. I guess the concern that was raised by Randy about the use of dichloromethane, particularly going forwards, is the NCI reconsidering that at all in the context of environmental consequences, and also perhaps access as well? Because it sounded from that in some places there may be problems with access to that. So using that in a procedure that all the different groups would be incorporating, it sounds as if that could be a bit problematic. So yeah, I mean, obviously the data suggests that mixture is perhaps the most effective, but as a standard procedure across a broad-based group of people, and thinking of the future as well, and that perhaps there may be even more emphasis to stop using that, it might be important to consider that.
Yeah, I mean, I can actually ask Barry O'Keefe what his thoughts are. I think that's actually a good point. And my guess is that the answer is going to be yes, there's an alternative. We just have to accept that it's not the same thing, and it's going to have limitations. But if that is good enough for the final purpose, then I think it would make sense.
Yeah, I think going forwards, because this group is looking to the future, and as you said, a protocol that it could be used by different groups in very different locations, it might be worth reconsidering the use of dichloromethane and finding out something that would be more environmentally appropriate and also perhaps more accessible to everybody.
Yeah.
Marcelino, any comment on that? Oh, yeah.
Yeah, can you hear me?
Yeah, hi, Christian.
Okay, hi. I just wrote down a question in the chat just in case. But well, yes, my concern was I'm going to moving on to greener chemistry, like supercritical, and I see lots of concern here, and I read about lots of groups that are moving into these techniques, you know, even in South America.
Yeah, so the question is, first of all, does everyone have access to those techniques, right?
No.
And second, from the NCI perspective, my understanding was that they wanted something that could be executed in a large scale and easy. So maybe that was part of the reason, but yeah.
I have a comment.
So how is sorry? Yeah.
Some groups use ethyl acetate, maybe it's less bad, and butanol also. Some groups use butanol. It's very hard to evaporate, but you mix it with a little bit of water. You can evaporate in a roundabout. Also, some people sonicate, you know, in methanol to break the cells, and then the cells release the content. But I don't know. This is better than the mixture of dichloromethanes and methanol, but we can try. And some people use these solvents that probably are less dangerous than dichloromethanes. So, well, I think that we need to standardize something and decide what we're going to use. This is the best solvent extracting, but it's not the best environmentally. And we need maybe find something intermediate, you know, to respect the environment and also to get a good extraction.
Yeah, that's right. So, Christian, what is Tamara using? Do you know? We can hear you.
Your microphone, yeah.
It's not working.
No, it's not working. You can write in the chat. You can write in the chat.
Yeah.
Usually she's using ethanol, and well, but she comes from a marine universe, so it's not exactly. Even so, it's not. It's different to what you showed just now with marine organisms. But
yeah,
ethanol
but
is like most common method, and we used to use dichloromethane, methanol, and water, usually doing like a sequence of extractions. That was when I was working at the lab many years ago, and that was an NCI's protocol at the time.
Yeah.
okay, I wanted to ask you also, Edgar, who also work on marine compound. What do you use in your lab, Edgar?
Hi, Vanessa. We are using CUC, erizos de mar, as biological model. Extract in celomic fluid some compounds, antioxidant, antimicrobial, from celomic fluid. These compounds have antioxidant and antimicrobial activity. Is very good for using in food for aquaculture organism.
Okay. Okay, thank you very much. I don't know, Don, I think I saw you were going to say something
No,
earlier.
no.
Yeah. No?
Soaking up all the information here.
Okay. Okay, I think probably some of you want to continue this conversation. I think it will be good if Marcelino and Antonio and who else wants to follow up, because I think probably Antonio will be good if we do an update of our first protocol, yeah, based on this presentation, to have like a new version of our protocol, yeah, so we can share with the new members.
Yeah, I think, but I think it will be important to clarify this TCM aspect, and I can email Barry O'Keefe to see if he has any insights, and if some of you can discuss this with other people, and we can put a protocol, some guidelines for a protocol together that would be easy to follow, right?
Yeah, I think the time is coming to an end, so I just want to make a last announcement. We are in conversation with the University of Costa Rica about having our next seminar in May or June in San Jose. So we will keep you posted on the dates and what is going to be our main focus. Yeah, I just received those news today from Costa Rica. They confirmed that they are going to be hosting our next seminar. Yeah, so those are very good news. And I don't know if anybody wants to add something, but I think then we are probably, Antonio, you want to say something, the last words?
No, no, just I hope this was helpful to everyone, or at least it's a first step to keep moving forward. And thank you for all your comments, all your feedback.
Yeah, and thank you all for participating, for being here.
Muchas gracias a todos. Nos vemos la próxima vez.
Muchas gracias.
¿Ya? Gracias.
Right.
Thank you.